Okay, so for question one, we've got 4 different viral genomes up here, and we're trying to use the information that we're given in order to determine the structure of the viral genome. And so we'll go through those real quick. So for virus 1, we're given the percentage of each one of these nucleotide bases, and we're also given whether or not it's affected by exonucleases or endonucleases. And we did some research to figure out that exonucleases, those go and affect the ends of the virus. And so if it's a circular virus, it's not going to have any ends, so it won't be affected by the exonucleases. It will only be affected by the endonucleases. So that gives us one of the pieces of information that we need. So looking at that, virus 1, because it's not affected by the exonucleases, we know that's circular. Virus 2 is affected by the exonucleases, so that's linear. Virus 3, same thing, affected by the exonucleases, linear. And virus 4, not affected by exonucleases, so that's going to be circular. So that gives us all that information. Then to figure out whether it's DNA or RNA, we're going to look at the percentage up here of the T's and the U's. Because DNA is the only place that we find T, and RNA is the only place that we find U. So that shows us that virus 1 and virus 2 are both going to be RNA, and virus 3 and virus 4 are going to be DNA. The last part to figure out double-stranded versus single-stranded is we're going to look at the proportions and the percentages of the letters that correspond. So we know that A is always going to go with U or with T, and that C is going to always match with G. So if this is a double-stranded virus, we're going to see that the A and the U, or the A and the T, are going to match up pretty close. So for this one, we see that 28% and 26% are pretty close. For C and G, the same thing. So we know that this one, virus 1, is going to be double-stranded. However, if we take a look at virus 2, our percentage of A is 34, and that doesn't match up with our percentage of U, which is 15%. Same thing with the C and the G, so we know that's single-stranded. We do that for virus 3, and that shows us that the A matches the T rather close, and the C matches the G rather close, so it's double-stranded. And for virus 4, they're going to be single-stranded because the percentages don't match. So that gives us the answer to number 1. Alright, so for question 2, it's to fill in the blanks. And we're going to go through these. Part A is looking for what's composed of 4 protein subunits and uses ATP to compact bacterial chromosomes. So that's topoisomerase 2 because it uses ATP and it has the 4 protein subunits. While a nucleosome contains 146 to 147 base pairs of DNA and 8 histone proteins. So we know that nucleosomes contain 146 to 147 base pairs of DNA. So that's obviously the answer there. For B, the DNA double helix, it was given to us that the DNA double helix is 2 nanometers wide. And then we learned that adjacent base pairs, they're separated by 0.34 nanometers in the DNA double helix. Topoisomerase 1 is what introduces positive supercoiling into DNA and cuts 1 DNA strand, whereas topoisomerase 2 introduces negative supercoiling. The origin of replication, so part D, the origin of replication for E. coli is called ORIC, O-R-I-C. And then for E, this goes back to the beginning. It talks about the Hershey-Chase experiments. So the bacteriophage, T2 proteins that were labeled with the radioisotope of 35S, so 35 sulfur is the radioactive isotope that was injected into that protein, whereas the radioactive isotope injected into the DNA was 32P, so 32 phosphate. And then for F, the sugar in uridine is ribose, while the sugar in deoxyphthalmidine is 2-deoxyribose. And that goes back to its name. So we look at the name because uridine ends with an ene in it. It's from ribose, and deoxy shows us that it's deoxyribose for that one. Ready?
Okay, so for question number 3, we have chromosome 14, which has 109 million base pairs, and chromosome 21 that has 48 million base pairs. And they go through translocation, so they're put together, making 157 million base pairs. So for part A, it's asking how long it will be in meters. So we know we have the 157 million base pairs, and for every base pair there's 0.3 nanometers, and then you just convert the nanometers to meters and you get 0.54 meters. And then for part B, it's asking how many nucleosomes are in it, and so you take your 157 million base pairs, and for every nucleosome there's 196 to 197 base pairs, and so you'll end up with 7.96 to 8.01 times 10 to the 5th nucleosomes, depending on which one you use. And then for part C, it's looking for how many H2A will be inside there, and so you take your 157 million base pairs, do the same calculation there, and then for every nucleosome there are 2 H2As, and so we end up with 1.6 times 10 to the 6th H2As. And then for part D, it's asking for how many radial loop domains there are, and so we have our 157 million base pairs, and then for every radial loop we have 25,000 to 200,000 base pairs, and so if you do the calculation with both of those, you end up with 785 to 6,280 radial loop domains.
Okay, so in number 4, you have a single circular chromosome, and this is how long it is for the base pairs. And so you know that you have 3.4 times 10 to the negative 4th micrometers per base pair. And so when you multiply those together, you get 1,500 micrometers. For number 2, it's wanting to know the diameter. And so if the circumference is pi times your diameter, we already figured out the circumference because in micrometers, because it's like taking the circular chromosome and stretching it out long. So that would be your circumference. And so you just divide what we found out here by pi, and you get 477.3 micrometers. So for C, it wants to know if one of the loops was stretched out, how long it would be in micrometers. So you use the same conversion factor as before. The 3.4 times 10 to the negative 4th is 1 micrometer per 1 base pair. And so you multiply it by these base pairs, let's cancel out, you get 17 micrometers. And then for D, it wants to know how many histones are found attached to the chromosome. And so you get 196 base pairs per 1 nucleosome, and then 8 histones per 1 nucleosome. And so when you multiply those out, then you'll get 180,000 histones.
Alright, so for question 5, we have all of these different structures, and for part A, it wants to know which of the molecules are nucleosides. So a nucleoside is just a base and a sugar. So it would be A, because it's just the base and the sugar. E is just the, E here is just the base and a sugar, and F is just the base and a sugar. Part B wants to know which are nucleotides, and a nucleotide is a base and a sugar with a phosphate group. So here B, we can see that phosphate group is added on there, as well as C, and as well as D. So for part C, we're looking for which ones have a ribose, and a ribose has 2 OH groups on the sugar. So C has 2 OH groups, E has 2 OH groups, and F has 2 OH groups. And then for D, it wants to know which ones contain deoxyribose, and so those only have 1 OH group, so that'd be A, B, and D. And then for part E, it wants to know which ones are pyrimidines, and pyrimidines are the ones that have 1 circle on their base. So this one has 2, that one has 2. So it would be C, E, D, E, and F.